I hate being last on the speaker's podium, but what I'd like to do is focus very precisely on systems medicine and talk about areas where I think we've really used systems medicine to move things forward. And that is talking about dynamics in the brain, and I'll give you an example in mouse and an example in humans, and then talking about some systems-driven strategies that we've pioneered here at ISB and show you how you can begin to change clinical practice. So the systems approach to medicine, as I indicated earlier, is really about the idea in a relatively short period of time you'll all be surrounded by a virtual cloud of billions of data points. And the real question is how we take these diverse types of data and integrate them together to make hypotheses about optimizing wellness and minimizing disease. And I think the issue that's really critical to understand is that in all large data sets, there are enormous signal-to-noise issues. And I think one of the fatal flaws in a lot of what's been done in medicine recently is people don't realize how much noise there is. That's why many published papers never, ever get repeated. And of course, as I showed you earlier, one way to take these types of data is to create what we've called this network of networks and then to be able to follow its dynamics. And I'll give you, as I said, some examples of where we've started to do this. One I've talked about before, it's been published, it's Prion disease in mice, but I think it really typifies exactly what we're going to be able to do in understanding the dynamics of disease. So this is a disease in which you inject activated prions into the brain, and in one strain of mice over a period of 22 weeks, the disease progresses to termination. And we analyze the transcriptomes at 10 different time points across that progression, subtracting away normal transcriptomes from their diseased counterpart to get the genes that are differentially expressed. And the really critical point here is initially a third of the mouse genes were differentially expressed in the brains, and they were statistically clearly differentially expressed. So the idea is there's an enormous amount of noise there, and how do you go about identifying signal from noise? One approach that we took was to look at a combination of 6 inbred prion strains of mice and to design them so that we could subtract away other aspects of biology that contribute to the brain transcriptome, eventually concentrating just on the differences that were related to neurodegeneration. And we did that, and I'd be glad to discuss what some of those combinations were. And we got more than a 20-fold enrichment down to about 300 genes. And then we could map these genes into the histopathology of the disease that had been done serially, again, across the progression period. The 4 major processes, prion replication and accumulation, microglial activation and inflammatory response, and then 2 different forms of neurodegeneration. So we mapped into this, into that, not only the transcriptomes, but 6 other parameters as well, and I won't really talk about that in detail because I want to stress the really important points. So one critical point is that the 4 major networks were actually sequentially disease-perturbed across the progression of the disease. And the importance of that is it's the proximal disease-perturbed network that's going to be the most key both for diagnostics and for thinking about therapeutics. And its identification gives you insights into the initial stages of the disease process and everything. The second point that came out is when we mapped the 300 genes into these 4 networks, 200 mapped in beautifully. The other 100 defined 6 smaller networks that showed similar kinds of dynamics. And collectively, the dynamics of those networks explain virtually every aspect of the pathophysiology of the disease. So it was really a beautiful example of how we can get down to some of the deep and fundamental understanding of this particular disease process. And I'll just say we've not only done that for prion disease, but we've done it for frontal temporal dementia and have fascinating results there that are quite distinct from their prion counterparts. And we're just beginning to do it with Huntington's disease now. And in fact, tomorrow, Terry Van Dyke will actually talk about a model of glioblastoma, or 4 models of glioblastoma, that mimic very closely the human disease and the dynamics and the insights that we've gained from that. Now, one of the really interesting questions is, can you really do dynamics on human diseases? And there is at least one possible way that you can do that. And it's one that we've studied human glioblastoma for. And that is, with a collaborator in Taiwan, we've actually identified 10 patients that successively had either 3 or 4 glioblastomas. They were treated with the 2 classic drugs for glioblastoma. Clearly, the drugs had a part of deriving or selecting this process. And these were ideal for looking at dynamics, for asking questions about the nature of genomic variation, about what drug selection did, about identifying the driver mutations, and so forth. And I'm only really going to talk about one of these things today. But I will say one of the challenges we face these days is the enormous amount of data. So for those 10 people, normal genomes and the cancer genomes, that is 13 trillion nucleotides of data that one had to analyze. And of course, one of the things we've done here at ISB is set up a production line for being able to deal with human family genomics and create a production line to be able to deal with large data sets like we see here. The most interesting thing that emerged from these initial studies, and we're only in the midst of finishing them, is that there were 2 really distinct phenotypes that were present in different tumors. One was a mutator phenotype, where the mutations really had to do mostly with single-base mutations and or deletions. And the second was a chromosomal instability phenotype, where we saw chromosomal rearrangement, we saw enormous epigenetic factor rearrangements and so forth, and interesting aneuploidies. And the 3rd and 4th tumors that we studied in this series were respectively of the mutator phenotype and of the chromosomal instability type. And here's just a circle diagram of the entire genome of these 2 patients. And all of the numbers you see on the most outer extreme represent the single nucleotide substitutions. So patient 3 is the mutator type, and patient 4 is the rearrangement type. And on the inner circles, you can see large deletions, rearrangements, and a variety of that. So here's what the first tumor looked like. Here's what the second tumor looked like. And if you can't see it well, it's just more of the same in each of these 2 respective tumors. And then the third ones were even more extreme. Now, the other tumors fell somewhere between those 2 different extremes. But the really interesting point is these are fundamentally different mutational mechanisms that are operating in the context of the progression of these tumors and so forth. And so we're thinking about what the drug selection process does. And the other thing we can do beautifully, and we have long lists of these, is we can distinguish beautifully driver from passenger mutations by creating for each patient a genealogy of the mutations and seeing what are the driver or the continuous mutations that have been a part of this entire process. And as I said, this was done primarily by Leslie Chen, who is now at Chang Chung Memorial Hospital in Taiwan with his colleague, Professor Wei. Strategies for systems medicine. We've developed two here, family genome sequencing. It integrates genetics and genomics and lets us identify disease genes and, frankly, wellness genes as well. And then taking a systems approach to blood diagnostics. So let's talk about family genome sequencing. The initial study we did that was a proof of principle was a family that had two normal parents and two kids, each with the same two different genetic diseases. And by sequencing the genomes back 3 years ago, we were quite confident we could reduce dramatically the number of disease gene candidates, and we did. We reduced it beautifully to 4. But we learned a lot of other things. One, by family genome sequencing, you could reduce the sequencing errors by anywhere between 70 and 90%. Number two, you could use the structure of family genome sequences to infer virtually complete and precise haplotypes for the children with respect to the chromosomes the parents had given them. And that's really critical for being able to ask the question, in the individuals with the disease phenotype, what are the unique shared haplotypes that are not seen in the normal controls? And if you just think about a simple family like this, that reduces the chromosomal search space by 70%, 75%. And we've had multi-generation families where the chromosomal search space is 0.1% of the search space. We have focused on a variety of diseases. We've done more than 1,000 genome sequences. As I said, we've set up automatic pipelines for taking advantage of family genome sequencing and so forth. And I'll give you just 2 examples of our focus on brain disease. So one is alternating hemiplegia of childhood. And what's really been beautiful about this is you can actually identify these disease genes in the context of individual families. So you can see that there are 5 families there for which we could identify disease genes. And then there were a whole series of sporadic individuals we looked at that all mapped into one of the 5 disease gene locations. So, and again, this was done in a relatively short period of time. A much more challenging analysis, of course, is looking at bipolar disease. And again, it's a disease that is psychiatric, it's diverse in character. We sequenced the genomes of 200 individuals from 43 different families, and we sequenced them for various delineations of bipolar disease. And I think the really important examples that we have now, and they lead us to the conclusion that if we double or triple the number of family members that we've sequenced, we can really generate an enormous amount of additional information, is that in a number of cases, the variants are associated with networks that are absolutely relevant to brain kind of function, like the voltage gate calcium channels and so forth. And I think across the population, there are going to be diverse influences that are going to modulate the genes, but if you have very large populations, you're certainly going to be able to drive the more important identify the more important driver genes. And it's really clear that family genome sequencing is really going to be an effective method for finding these. I'll say we've done probably close to 100 genes for Huntington's disease. And it's an interesting one because you know the disease gene, you're looking for genes that modify the effects of the disease gene. And again, we've got terrific candidates, and we're utterly convinced that sequencing additional families is really going to be transformational in that particular area. So we've got a wonderful group here at ISB, led by Jared Roach and Gustavo Glussman, that have set up the family genome sequencing, and we've had a lot of diverse collaborators, including John Kelso, who contributed most of the 200 patients whose genomes we've sequenced. And the final area I'm going to talk about, I think, is the one that is going to have the most impact immediately on clinical medicine. And that is systems diagnostic, making blood a window into distinguishing health from disease. And as you all know, there have been many papers written on diagnostic markers for all sorts of diseases. And they've all been of the general framework of, let's look at the bloods from normals, let's look at the bloods from diseased. Gee, we see lots and lots of differences. The trouble is 99.9% of the differences are noise. So how do you pluck out the signal? And I would argue that very powerful ways come out of systems approaches for plucking out the signal. So our belief is blood is the key organ because it bathes all organs and they secrete proteins into it. Some of the proteins they secrete into it are organ-specific, that is, uniquely synthesized, say, in the brain. And the reason those are so important is they provide a molecular address to say, look, changes in the brain organ-specific proteins have occurred, and actually those proteins even tell us about the networks that are involved in the corresponding affected regions. We think longitudinal analyses are key, multi-parameter analyses are key, so you can sense multiple different networks, and you want to do it as quantitatively as possible. And we've had terrific luck both with proteins and with microRNAs. And the protein approach that we've taken is using targeted proteomics to make beautiful SRM assays for hundreds of candidate proteins. And I'll take you through one process where we've sorted out beautifully noise from signal. And that first case is a non-brain case, but it's the one we've come furthest. Collaboration, well, all the work was done at Integrated Diagnostics by Paul Kearney and his colleagues, and we've collaborated, Nathan and I, kind of conceptually on this. But it's really been a wonderful study. So the idea is this. There are 3 million nodules in lungs a year, and the big question clinically is, what do you do with that nodule? And the answer is there are a bunch of different things you can do. You can wait, you can do a PET scan, you can do an aspiration or a biopsy, or you can do surgical resection. And what's interesting is close to 40% to 50% of the surgeries that are done on benign nodules, and hence you put the patients at risk and you cost the healthcare system lots and lots of money. So the question is, could we take an approach that allowed us to distinguish benign from neoplastic nodules in lung cancer? And the approach that we took, I'm going to outline because it gives you a beautiful idea of how you start sorting out signal from noise. We started with 371 targeted proteomics assays for lung cancer. Some of the candidates came from comparing normal lung cells with their cancer counterparts and seeing which secreted molecules and which membrane molecules were different in those two cases, and adding those to the candidate list. And of course, we searched the literature. What we found, and another constraint for all 371, is they had to have been reported as identified in the blood. And we found by the initial studies that we could see just a little bit better than 50% of them. We saw 190 that were reliably detectable in the blood. So what we did was to take those 190 and to examine plasma from 72 patients with neoplastic nodules and 72 with benign nodules. And then what we did was to rank order those 190 proteins according to who could make that call most effectively. Then what we did was to select the best 30-some, and we created 1 million panels of 10 proteins randomly chosen from those 32. We rank ordered all of those panels with regard to their diagnostic ability. And then we asked the question, which were the most successful cooperative proteins, that is, were found most frequently in the most effective panels of 10? And we came out with 13 that looked quite good, were highly cooperative. And then what we did was a validation study where we took 52 independent patients with cancer nodules and 52 with benign nodules from the same sites, but from a fourth site as well. And in fact, the results in the fourth site were even better than the discovery and validation sites in the first sites. So this put us in a position to think about, or integrated diagnostics in a position to think about doing a soft commercialization of this product starting next month, which is really exciting for someone who's been doing diagnostics for a long time. So the test has about 90% negative predictive value. That means we can identify 9 out of 10 neoplastic nodules very beautifully. And it has 44% specificity. That relates to how many benign nodules you can see. So it means we can eliminate close to half of the patients for these surgical procedures. And if we do a plot of benign and cancer nodules, red and blue respectively, with their sizes indicated, and map that onto the smoking history of the patients, you can see at this 40% level, to the left, 90+% of the nodules will be benign. And those are the patients in a rule-out test. We can say, look, you don't need to undergo surgery. You do need to undergo watchful waiting, but we can tell you 90% of you will be benign in nature. And I didn't stress it before. If you do the calculations of the cost of procedures and how many are carried out and so forth, my calculations, these aren't integrated diagnostics now, are that this test ought to save the healthcare system between $5 and $10 billion, depending on exactly what your assumptions are, $5 and $10 billion a year. And again, remember, we're talking about 3 million nodules per year in this assay. The other really exciting thing is 12 of the 13 proteins map into 3 biological networks that are intimately associated with lung cancer. So this means not only do the markers give you beautiful pattern recognition for benign as opposed to neoplastic, they give you a window onto the progression of the disease. An incredibly exciting opportunity. And this is why you've got to filter away noise from signal. And when you do, you're in a really uniquely powerful position. And the last example I'll give you is using the same approach, and I'm going to talk about it a little less because we aren't as far along, to look at soldiers from Afghanistan that have post-traumatic stress disorder as compared with their normal counterparts. One of the key points that we've done is we've identified close to 200 brain-specific blood proteins that we can detect with targeted proteomics in the blood, and they map into virtually all of the major general functions of the brain, and I've indicated that in the slide as you can see it there. What's nice about these brain-specific blood proteins is, as you'll see, they're great for disease diagnostics, but they're also great for looking at normal biology in the brain, following development, following physiologic responses, looking at the process of aging, other kinds of things that you might be interested in. PTSD, as you know, is a major problem in the normal population. 1.3% of our population has really severe PTSD. And in fact, if you look at the soldiers coming from Iraq and Afghanistan, the estimates range between 5 and 25% because psychiatric diagnoses are so poor. So we decided if we had a quantitative assay, that really could be transformational. And to cut a long story short, we've looked at close to 50 normal plasmas and 50 plasmas from soldiers with PTSD. And the sensitivity specificity in this discovery phase, we haven't done validation, is really spectacular. It's in the low 90% or so for a panel of biomarkers that lets us assess this disease. And I will say, as an added measure of interest and complexity, the panel is made up of a mixture of proteins and microRNAs. The one protein that is most dominantly successful maps specifically to the amygdala, which is one of the 3 regions of the brain that is most profoundly affected by PTSD. So we're ready now to take the next step forward on the validation studies. But for me, what is really exciting is we have an approach now that will not only let us finish up with PTSD, we're moving directly to traumatic brain injury. That's athletics as well as the military. But I would argue that we can use these approaches for neuropsychiatric diseases like schizophrenia, bipolar disease, and autism. And I would argue we'll be able to do 4 things with these biomarkers. One is early detection. The second is follow disease progression. Third is the stratification of disease into different subtypes. And the fourth, for the first time, we'll have a quantitative means of following a response to therapy, which for a lot of these neuropsychiatric diseases is absolutely critical. So this has been a collaboration with a group funded by DOD from the Army and from Santa Barbara and from New York University. So I'd like just to close by saying I've only talked about a few different things here, but I think this systems medicine really is today beginning to transform healthcare. And I think it's actually going to do several things. It'll improve the quality of healthcare. It'll reduce the cost of healthcare. And I think it's actually going to promote enormous innovation in the ability to create new companies, because I think there's going to be a transformation in the healthcare industry in the next 10 years. And if you don't adapt yourself to these systems opportunities, you'll be a dinosaur. And there'll be a lot of dinosaurs, so there's a lot of opportunity for young, new, agile companies that are optimized for the opportunities of systems medicine. But basic fundamental mechanisms, the family genome sequencing, the identification of disease genes, making blood a window into health and disease. I didn't talk about it, but we have 3 really terrific ways for thinking about how to stratify disease into its subtypes so you can have an impedance match against therapy. And the final thing I would say is the idea that patients all have to be stratified in accordance with their response to environmental signals, such as drugs, infectious agents, or a variety of other things, is really critical. It's every bit as important to treat the individual as a quantized unit and put them in similar populations. It doesn't mean we're going to have a different drug for each individual. It means you're going to look at the individual and classify them into groups where you can optimize treatment. Thank you very much.